Okay, so that's a little bit about what is a virus and its basic structure, but what about its pathology, what about its biology? How does it actually do what it does to plants and cause disease, which is probably what we really care about. So, the basic life cycle of a virus. Infection first. So it's actually pretty crude. When we go and talk, or when we start talking about fungi and some of the other more specialised pathogens, they'll actually have, some of them at least have quite specialised infection structures that can penetrate cells and do some pretty sophisticated infection processes. Viruses are pretty simple. They can just got to get in somehow. Now they've got some significant barriers. So here's a plant cell, here's a leaf. You've got a cell, you've got a cell membrane, you've got a cell wall, then you've got this waxy cuticle on the outside. Now that's like Mount Everest to a virus. It's got to somehow get through there and into that cell. And it's got to get through there into that cell, and that cell's still going to be alive at the end of it. Why does it need to be alive? Anyone?
So that it can reproduce the virus.
Absolutely, because it's a cellular parasite. If it's a dead cell, it can't. It's bacteria might like to eat the dead stuff. Viruses need a living cell to replicate. So it can't replicate outside. It needs help to get in. So they have no specialised structures. It uses micro wounds. So wounds that will actually penetrate and allow the virus in. Now that can be mechanical. So leaves rubbing against one another will cause damage, abrasion that will heal. But those small openings can allow viruses to get in. So some viruses do specialise in what we call mechanical transmission, and they get in by mechanical means. So there's millions of virus particles in this leaf, it rubs against another one, and a few manage to get into the leaf, a few cells of the other leaf, and away it goes. More commonly, however, that they use things like sucking insects. So here we get our entomology back again. So aphids, thrips, whitefly, particularly those major ones, but others. There are some beetles, there are a range of different insects that can specifically transmit viruses. Why do you think something like an aphid is so good at transmitting a virus? What does it do? How does it feed? Come on, you just done entomology. They have stylets, they've got these specialised mouth parts, and they pierce into cells and suck the cell contents. They don't chew it. They're not like a grasshopper who takes a big hunk out of it and kills the cell. These guys pierce into cells and start sucking cell contents. Particularly when they first arrive in a plant, they go probe into all these epidermal cells to see whether it likes the taste of it. And in doing so, it can be picking up viruses and transmitting viruses because they're actually providing essentially like a hypodermic needle, sucking out the virus, spitting it back out again. It's not quite as simple as contamination, decontamination, but it's vaguely that sort of way. There is actually specific associations, but that's how it works. It sucks it up and spits it out again. So it gets in. Once it gets in, it needs to replicate. So they use the host enzyme, the host protein synthesis machinery, so the host ribosomes, to read the nucleic acid. So it gets in there, a virus gets in there, strips off its protein coat, releases its nucleic acid, which gets grabbed by the ribosomes, gets read, and they start producing virus proteins. Some of those proteins will be enzyme proteins that allow the virus to replicate its own genome. So it quickly makes what we call replicase enzymes that actually make millions of copies of the virus genome. It makes coat protein and then sticks them together and makes, so one virus becomes a few billion virus particles. Once it makes the coat protein, nucleic acid, as I said, it joins it together, it coats up again, and you've got new particles. Viroids don't need to make proteins. They actually just replicate themselves a bit like They call it a rolling circle model. It's a bit like a toilet roll. So here you've got your template. Here's your viroid. And it starts copying it. And then keeps going, and you've got... It's a bit like unrolling a toilet roll, and then you rip off the perforations and reform them into circles. That's kind of how it does. So this thing keeps going round and round and round. It keeps making copies of it and reforming new copies. Slightly more complex because it's got to go from positive to positive strands, but essentially that's how it works. So you use the circle, just keep copying it, copying it, copying it. A bit like a printing press, really. I keep dropping this. Okay, so it's got in the cell, it's replicated in that one cell. That's pretty useless for the virus. It's good if that's all it does for an agronomist, because one infected cell is not going to worry you in terms of the plant, but it needs to get out of that cell. It needs to move through the whole plant to actually cause disease. So intercellular spread, we've already mentioned this. It actually moves between cells through the plasmodesmata, through these cytoplasmic connections, until it hits the vascular system, and then it scoots up and down the phloem. Why the phloem and not the xylem? Anyone got an idea? What's the difference between the phloem and the xylem of a plant? Which one's alive? The phloem's alive. The phloem's actually living. The xylem is dead. So a virus that's an obligate cellular parasite that needs to live inside a living host is not going to survive or replicate or move in the xylem. Saying that, there's at least one weird virus that does, but we'll ignore that one. So here's an example of typical spread. So we got an aphid or something that drops off a virus and infects that leaf right there, starts to replicate in that cell, starts to slowly spread from cell to cell to cell, gets into the starts to invade, much or most of the leaf itself, but gets into the vascular system at that point, and then starts moving more rapidly through the phloem. Now it tends to go to the top and the bottom, probably to the bottom first, so getting out at the bottom and into the roots and starting to spread out into the new invading roots. Goes to the shoot, starts spreading out, invading from the meristem out into the new leaves, and then can slowly work its way down and start to invade some of these older leaves. There might be some really old leaves that never get infected. Why? Because they might be metabolically pretty well stuffed. They're actually big, they're fully expanded, they're actually not doing much. Viruses need the cell metabolism to be active to replicate. So if they get into this old cell, this old leaf, and it's actually stopped replicating because it's starting to senesce or it's not doing much, it can't replicate itself. So it gets out into the growing tips. That's also why when you're looking for a plant virus infection in a crop, you'll often be looking in the newly formed leaves, because that's where the systemic infection first goes and where you're going to get the most symptoms. So these young leaves are going to be the really distorted ones, because from a very early age of development they've been infected and then they start to show symptoms. How do they cause symptoms? They can cause an amazing array of symptoms, and it's essentially by screwing around with the host genetic gene expression and metabolism. So they're direct competitors. They actually are parasitic on the host, so that they kind of steal the ribosomes from the host, and so they go make virus proteins, don't make your proteins, and they can actually hijack the ribosomes and force them to make virus proteins rather than host proteins. So that's actually a competition. But they also can switch off gene regulation in the cell, and that can cause some pretty wacky sort of changes in the biochemistry. And it can be anything from no symptoms through to plant death. That's unusual because a virus doesn't want to kill its host, but there are some virus-host interactions where it will do so. Yeah, that's a nice quote. They can do anything. Pretty well screw up most of the way the plant operates itself. What sort of symptoms? We've kind of seen some of these before, but you'll get hyperplasia, stunting, abnormal development, that's quite common. So things like stunted growth, things like instead of big tubers, little tubers, that's a viroid. Big plants, little plants. Big, that's probably a pumpkin or a zucchini, and a small one that's infected. Or distorted growth, again, where you've got puckering, you've got these reduced lamina, you've got leaf curling, all related to distortions in the growing tip. So you've actually got some cells that are growing fast, some cells that are growing slow because they're virus infected, and that ends up with this puckered growth. So how does it happen? Competition, reduced photosynthetic capacity obviously causes stunting. If you reduce the amount of leaves, obviously they can't produce as much sugars by photosynthesis, and that's obviously going to lead to stunting as well. You're going to damage the roots. This goes systemic through everything, so it'll actually potentially damage both the vascular system and the root tissues, and reduce assimilate uptake. It can affect the way you distribute carbohydrates. A virus infect. What's the best example? There's fruit trees, things like prunus species. When they get virus infected, they can actually switch whether the sugars primarily go to the new growth or the second year's growth. So, and I'm going to get around the wrong way again, but things like an apricot, I think it normally fruits on second year wood, but when it's virus infected, it fruits on first year wood. And can affect nitrogen fixation. Can also play around with hormones and change things. You can get mosaics where you've got dark green tissues that actually have less virus in them growing faster than light green tissues that have more virus in them growing slower, and you end up with this uneven growth. And all of this is actually related to the way the virus actually defends itself against plant defense processes, genetic defense processes. There's actually a natural defense process in all organisms that are actually active against viruses, and it actually finds out, oh, I've got a virus, I'm going to try and switch it off. Well, the virus doesn't sit there and go, oh, gee, you're going to kill me. It's actually evolved processes to actually fight back. And so the mosaic is actually the foreground of the battleground of the virus, of the plant and the host. So the dark green cells are where the plant's winning. It's not completely virus-free, but the plant is actually successfully repressing the virus and has got its defense system switched on, and it's going, you're not going to come here. The light green cells are where the virus has won, and the virus has switched off that defense process, and it's actually multiplying like crazy. And that's why this mosaic is really a representation of that battle between parasite and host. You can get necrosis. Uncommon because dead cells don't replicate viruses, but here's some necrosis, not full necrosis, but necrosis where the virus is moving out of the veins. Again, some fruits. Those are both tomato spotted wilt virus. And the reason why tomato spotted wilt virus can have hosts that cause necrosis is because it's got such a huge host range. It's got some good hosts and it's got some bad hosts. So it's the good hosts that replicates normally and doesn't kill them. And then it sometimes gets into some hosts and it does. So obviously that's a dead end host for it, but for the agriculturalist that's pretty nasty because you can actually have your plants dead. So tomato spotted wilt virus can kill tomatoes, for example, which is pretty nasty. It's a very nasty disease of tomatoes and capsicums worldwide, and particularly in Queensland here. It's not necessarily its preferred host, but it's just a pretty good one at knocking them off. Chlorosis, however, is very common. So viruses, again, they're a cellular parasite. They affect the metabolism. One of the first metabolic processes to cark it is chloroplast development and chlorophyll production. So it can reduce chloroplast production, and then it can actually then decrease chlorophyll synthesis. and you'll get some pretty patterns, I think. You know, ring spots, some of those can look quite nice. Not if you're trying to grow it as a crop. This peanut with a very pretty disease. It just doesn't produce any peanuts. There's a rose bush with a virus. The mosaics. You can also get colour change in flowers, which we tend to call colour breaks, and that might be different pigments. So in this instance, here's our tulip again with tulip breaking virus. Rather than chlorophyll we're switching off, we're actually, in this case, switching off some of the anthocyanins. So the red pigment is switched off, and the underlying colour is yellow. So normally you wouldn't see the yellow because it's mostly red. Then you switch off the red, and all you see is the yellow behind that, and so you see these yellow streaks. You can sometimes get excessive growth, these weird little emanations and funny puckerings and growths called hyperplasia. It's not that common, but again, it's partly related to the fact that these viruses, the way they interact, and they start switching genes on, switching genes off, playing around with internal hormone levels, can really cause some amazingly weird changes to the plant physiology. So, our virus has got in, it's replicated, it's moved from cell to cell. It's actually caused disease. So that plant is now cactus. But for that virus to be effective and to be a problem for your agricultural production, it's got to get from that one plant in the corner to the rest of the plants in the field. So how does it get from plant to plant? Well, they're fairly clever. They've evolved a whole bunch of different means. Firstly, they can associate themselves with the plant propagules. So that makes sense. You've infected a plant. Why don't I just hang around in the plant seed, and then when you plant the seed again, I'll be there already, and I don't have to leave my host. So that some, but not all viruses, can associate with seed. Vegetative propagules is more common, so things like potato tubers, things like tulip bulbs, things like rhizomes, things like ratoons, sugarcane ratoons. They are all vegetative propagules. They are very commonly infected. So if you've got an infected potato plant, the chances are all the potatoes are going to be infected. You harvest those, you replant them, your crop's infected. Some of them will also associate with pollen. They tend to hang around the outside of the pollen grain, so obviously it's short-lived, but that can be a really useful way of moving from plant to plant. So for instance, a lot of the fruit trees, things like your prunus species, many of the important viruses actually associate with the pollen. And so that's tricky because obviously you want pollination to happen. If you don't have pollination, you don't get fruits. But pollination is also the process of virus infection. It's a little trickier than that because the pollination process needs help for the virus to actually infect. In fact, you actually need some thrips, pollen-feeding thrips in the flowers to actually act as little virologists and inoculate the flowers for infection to occur. But it makes it tricky. Obviously, you can't spray out your bees to avoid virus transmission because you won't get any fruit. So it's an interesting scenario where management becomes tricky. You can also get mechanical spread. Now, virologists are really good at grinding up leaves and rubbing them on plants like we have here to maintain our virus cultures, and that's great, but it can happen in nature as well. So there are some viruses that are really well adapted to mechanical transmission. They tend to be really high concentration, they live in the outside cells of the plant, and when they rub on each other by plant-to-plant contact, they transmit. So things like tobacco mosaic virus we were talking about as the first one sort of characterised, that's a classic example of one that's spread that way. So we don't grow tobacco in Tasmania anymore, but we used to. And tobacco mosaic virus would have been very common, and it can, big fat leafy leaves that rub against each other in the wind, and transmit the virus. But you can also transmit it by grafting, pruning, grazing, harvesting. So anytime you're actually cutting the plant, you're releasing sap, and then you move on to a healthy plant, there is the possibility of transmission in that manner. And we've got many virus systems, particularly in perennial horticulture, where obviously grafting is important. You can be grafting new budwood onto a rootstock, and if the budwood is infected or the rootstock's infected, you're going to be transmitting that virus as you go. And that, for instance, is a very common way for a lot of the apple and stone fruit viruses to move around. You might also get things like this. This is a root prune. So these are two roots crossing one another, and they're actually touching enough, and they actually form vascular bridges and actually linking. So it might actually also be possible, again, in these perennial systems, for plants to be linking one another by vascular connections through their roots. There was a great example. This was not from a virus perspective. Well, actually, no, one from a virus perspective. We were doing some work in hops, and hops are interesting because they've got an annual shoot and they've got a perennial root. So this big root system underneath. And the virus was moving from plant to plant to plant down the row. So we were wondering, is there any vascular connections? Is there any root grafting? So went in there and injected glyphosate into one plant, and you watch all these neighbouring plants start to yellow. So yes, it was definite transmission of the herbicide from plant to plant to plant down the row, which obviously is a potential mechanism for moving viruses too. Vectors are really important. So insect vectors, nematode vectors, some fungi vectors can transmit viruses. So whiteflies, thrips, leafhoppers, aphids are really, really important. As I mentioned, these guys are important because these guys have got wings, so it's pretty easy for them to move from plant to plant. They can actually travel over long distances and carry a virus in that manner. They also tend to feed by stylets, where they penetrate into a cell, they suck the cell contents, they can become contaminated with the virus, and then they can fly off and deposit that virus somewhere else. Importantly, the association is actually quite specific. It's not just contamination, decontamination. There's actually recognition. So the virus actually recognizes the vector and binds to the vector's mouthparts, or the gut linings, and is carried in that manner, and then when it spits out into the next plant, it transmits it. So not every aphid will carry every single virus, but there'll be some specific aphid virus associations that actually allow that transmission to occur. Really important though, because managing spread, as we said, we're all if you're trying to avoid the disease, you're trying to avoid infection in the first place. If your infection's coming from out there and coming in by a mass movement of aphids, that becomes really tricky. How do you control that? By the time they've arrived in the crop and they've fed in the crop, they've transmitted the virus, then you spray them off, too late. You've already got infection. You might have controlled the aphids, but you haven't controlled the virus infection. It becomes quite tricky. So that's how it moves, and then it has to survive between hosts. So how does a virus survive? It can survive in the seed, in the tubers, in the corms, in the plant propagules. That's a good way. It can survive in a perennial host, obviously. It might jump into a different host species. So maybe this is chickweed. So for instance, cucumber mosaic virus, which has got a huge host range, causes major problems in lupin production in WA. But lupins only grow, well, they don't grow over summer obviously, they only grow in the winter period, and then you have no lupins. So the virus has to survive from season to season. Now it does associate with the seed, but it also jumps into weeds like chickweed and can infect those and sit there and waiting for a new lupin crop to appear, and then they can jump back into the lupin crop. Viruses can also associate, these are nematode cysts, and some viruses that are transmitted by nematodes can actually sit inside the nematode resting structure for years, waiting for a new plant crop to come along. So making the rotation somewhat difficult depending on how long the nematode survives for. Okay, to summarise, these guys can cause some pretty nasty diseases, and one of the main reasons is because their control is really difficult. You can't eliminate them, you've got to stop them happening in the first place. That is why they become so important. They're less devastating, they don't tend to kill things outright, but they tend to cause major yield losses, major quality losses, and you can't, once you've got them, you can't get rid of them. Their biology, they're subcellular, obligate parasites, they're quite simple, but they have a really complex association with the host, much more complex than most other plant pathogens. They a unique replication strategy. It's important to know what we're dealing with so we can manage it. So we need to know the type of virus we've got, the specific species of virus we've got, so then we know what aphid or what thrips or what vector, or maybe it doesn't have a vector, maybe it's mechanically spread. So we can understand its biology, we can understand how it moves and how we can intervene. Is it associated with seed? Is it not associated with seed? What's its host range? Does it live in that weed or can it not get into that weed? The more we know about it, the more we can do about it. And they can cause an amazing array of symptoms. Rather than just killing things, they can cause some amazing changes to the way plants grow. For successful infection, it's got to get into a cell, it's got to replicate, it's got to move. And then it's got to get from plant to plant. And obviously that's important for survival, but also important, otherwise we don't have an epidemic that we care about. It's just, unless it's your mum's favourite pot plant, one dead plant is not going to cause a huge amount of grief in a field, but a field full of dead plants or a field full of infected plants will. Okay, any questions? So, what we want to do, I've actually got 3 case studies there to go through. One I'm going to do, and then we'll see how we go. We might have time for a second. But they're there for your information to see some examples. So let's get an example. Let's do this one. So I just want to go through some real life examples of a virus disease in an important crop, how it works, and what we do about it. So we've done a little bit of the biology, now let's talk about practicality. So this is a really important disease in Tasmania, it's a really important disease in small grain production worldwide. Barley yellow dwarf. It's a persistent virus, it's an aphid transmitted virus. It causes major yield loss. It doesn't necessarily wipe out the crop, but can cause some major yield loss and really important, particularly in barley. It was originally thought of caused by one virus called barley yellow dwarf virus. So the viruses tend to be named after the symptoms and the host they were first described in. So barley yellow dwarf virus. But in the last probably 15 years, they discovered actually we've got 2 completely distinct viruses causing exactly the same disease. So there's another one they now call serial yellow dwarf virus that is genetically distinct, but does pretty much the same thing in terms of disease. Management's the same, but it's actually a different virus. Anyway, barley yellow dwarf, the disease, been around for about 50-60 years, been recognised as being a virus disease. Obviously the disease has been around longer than that, but that's when they first recognised it as a virus disease transmitted by cereal aphids. And it really is one of the most important diseases, if not the most important disease of cereal grain production. Particularly nasty in oats and barley, more so than wheat. Wheat is less affected, but oats and barley tend to be quite severely affected. It does invade quite a wide range of the Poaceae, so it also gets into ryegrass and other grasses and other crops too, other grain grass crops. Complete crop losses can occur, but that's unusual. It's usually around about 10 to 15% yield loss, which doesn't sound terrible, but you're talking about small margins in grain production, and that can be enough to lose your entire profit. So what does it look like? The symptoms kind of look a bit like nutritional disorders, and there's a reason for that. This virus lives inside and is restricted to the phloem tissues of the plant. So it doesn't actually get into the mesophyll cells, doesn't get into the leaf cells per se, it stays in the phloem of the plant. So it upsets the movement of photoassimilate through the phloem, and thus it really does look like a nutrient deficiency because it's really kind of causing that. If you put more fertiliser on that, of course, it's not going to make it any better, however. So you get yellowing and reddening in the affected plants. You get quite severe stunting, and that very much depends on how old it was when it was infected. So if you get a young crop infected, it'll slow the growth right down. You'll end up with essentially no yield at all, and it'll be horrible, stunted, and terrible. If it's infected quite late in the season, it might not cause a yield loss at all. It might just be superfluous because the plant's already grown, it's already started to fill its grain. Doesn't really matter. Plants can die, infected plants can die if you stress them, water stress them for instance. Not that common. The virus themselves. So these two viruses are members of the Luteoviridae. Luteo is Greek for yellow, so their virus is the virus family that tends to cause yellowing diseases in plants. The Luteoviridae all live in the phloem and all cause that sort of damage to the vascular tissues. They're all little round bubbles, they're all little icosahedra, they're all single-stranded RNA viruses. They are phloem limited, so infection is only in the phloem cells. So that's the phloem and the companion cells, but not the cells beyond that. It has not got the capacity to move into those and replicate in those cells, which is kind of interesting. And I kind of said that already. Why they discovered they've actually got two distinct viruses was when they started doing all these genome sequencing stuff. So they had all these different strains. They knew they had different strains of the virus. They had slightly different biology. They were spread by slightly different aphid species, but they all caused the same disease, and they all thought they were just strains of the same virus until they started sequencing the genomes of these viruses, and they discovered the barley yellow dwarf strains, and there's a couple of them, have a structure. These boxes here are the genes. Have a structure that looks a bit like this. So you've got these two make the polymerase that helps replicate the RNA, help replicate the genome. This is the coat protein. That's the actual protein that goes around the outside. This can sometimes make what we call a read-through protein. And this bit here is the bit that recognises the aphid. It's important for transmission. This bit here helps it move from in and out of the companion cells. So it's a movement protein. So that was the basic structure they all knew. And then they found these other strains and they went, yeah, this bit looks the same, but this is completely different. We've actually got a silencing suppressor, a defense gene. We've got a different type of polymerase, and the sequence similarity is it's completely dissimilar. So what they've discovered, or what they hypothesize, is that in evolutionary past, this virus is broken in half and taken this replication part from a different virus and stuffed it onto the luteo part of a luteovirus and created a brand new virus. this family, the Luteoviridae, are really good at doing this, what we call recombination, genetic recombination, where they can break themselves, recombine with a completely different virus and create a brand new virus. We're actually working with one of these at the moment in affecting peas, and it actually has a break point about here. And what it's done is this bit's normal, but it changes the read-through bit. And if you remember, I said that's the bit that's important for finding and recognising the aphids, the aphid vectors. So what it's done is this virus has got a huge geographic range from all the way up in Queensland right the way down to Tasmania. Those aphids don't exist, the same aphids don't exist in Queensland as exist here in Tasmania, the ones that are spreading it. So what we've got is it's adapted itself to the Queensland aphids by chopping off and changing this bit up north and having a completely different component down south to associate with a completely different suite of aphids. So very clever in terms of how do you expand your geographic range when you're completely dependent on an insect that has a specific environmental boundary. Change yourself and find a new insect. So we've got strains. We've got strains of barley yellow dwarf, we've got strains of cereal yellow dwarf, and they tend to be spread by different aphid species. So CYDV, the RPV strain, is spread by Rhophilosiphum pardae aphid. The MAV strain is spread by Macrosiphum avineae. The PAV strain is spread by both. They can vary in their type of disease, how severe their disease is when you put them into an oat system. But we've got strains, and we've got strains that vary. They vary slightly serologically as well, but they also vary because of the vectors that spread them and how severe they can be. Where do they live, or what can they infect? They've got quite wide host ranges within the grasses, within the Poaceae. So barley, oats, wheat, triticale. They do get into rice, but they're not a big problem there. They do get into corn, it's not a big problem there. Sorghum. Ryegrass, you don't see the symptoms in ryegrass, but it might, the belief is, particularly in the perennial ryegrass, that actually is one of the reasons that reduces its longevity, that actually stops its perenniality, because it stresses them and then water stress comes in and actually they die off and you have to resow them. So it can be quite important even in pasture systems. As I said, these are spread from plant to plant by cereal aphids, one or two different species. The association with that aphid is very close. So I kind of suggested before, whoops, this strain is spread by this aphid, not by this one. So it's a very specific association. This component here is quite specific. It's saying, I recognise Rhophilosiphum pardae, I don't recognise Macrosiphum avineae. So these viruses tend to be very, very specific, and part of that is because the way they actually transmit, the virus actually goes internal in the aphid. So you've got your aphid. It sucks up the virus. It goes into the gut system, and actually at the gut system here, it actually buds through the gut membrane. It gets into the hemosyl, or the blood system of the aphid. It doesn't replicate. It's not an insect virus. It can't replicate in the virus, but it does internalise. Then it gets carried. There's host proteins that help it, and it carries it to the salivary glands, and again goes through a membrane there. And once it's in the salivary glands, it can then get spat out into infected plants from then on. So it's complex. You've got at least two membranes that it has to recognise and actually pass through. That's where the vector recognition is incredibly important. But once it gets in there, once that's infected, it's then infected for life. So that aphid can run around and bite on as many plants as it likes and transmit the virus to as many plants as it chooses to feed on. So anyway, a few aphid vectors. Transmission is persistent. By this, it means that once it's acquired, it can transmit it for life. There are other viruses that are what we call non-persistent, and they actually tend to associate with the mouth parts only. They're picked up and they transmit it, but they can be lost. You can lose them off the mouth parts, and then the aphid might be free of the virus from then on. Once this guy's infected, it's infected for life. So it's acquired. It needs a long feeding period because the virus is in the phloem. So it actually needs to suck it up from the phloem. So the way aphids work, they... Stop dropping it. Here's my leaf. The leaf cells. The aphid initially feeds on these initial outside leaves and goes probe. Quick probes. Do I like the taste of this or not? The virus is not there. Remember, it's a phloem virus. The virus is down here in the phloem. So it probes for a while and then eventually settles down, and then it puts its stylet into the phloem and feeds from the phloem because that's where it gets most of its nutrition from as an aphid. So it takes about at least an hour, half an hour to an hour before it starts phloem feeding. At that point, it can obviously pick up the virus. There's then a latency period. And that latency period is the time it takes the virus to go here, to go here, to go here, to go all the way through to here. And that can be days. So it's not an instant transmission. It's not a suck and spit. It's not a contamination, decontamination. This takes some time. And then it's transmitted. Once it's in the salivary glands, it can be transmitted again. Once again, it can take a prolonged period because it has to spit it out into the phloem, not into the epidermal cells. So again, it needs to settle down and feed for a long period of time. Why does that become important? What was I saying now? Particularly important because for control, you've got a few windows of opportunity here. Because it takes a while to pick it up, you've got a period of time before it can transmit again, and then it takes a while to transmit. Insecticide treatments can be effective because you can see you can have like a pre-plant insecticide on your crop, and then aphids come in, they start to feed, and by the time they pick the virus up, and before it's actually gone through this whole latent period, the aphid's dead. So you actually have effectively stopped acquisition before transmission. If we compare that to these ones that are non-persistent, that actually only associate with a stylet, they can be transmitted in seconds, and insecticides don't work. Anyway, let's go on. These viruses cannot be transmitted through seed, so the seed is always clean. Why is that? Because seed does not have any vascular connections to the mother plant. So there's no phloem connections from the mother plant to the seed. There actually is physically separated when you form seed. That's plant physiology. So there's a virus that's stuck in the phloem doesn't have any mechanism to get into seed. So seed is always clean. So it can't survive in seed, it can't be planted with seed, it's got to come from somewhere else. And the virus reservoirs, where the virus sits, is in all of those grasses and other cereal crops in the environment. So you're planting a barley crop in Tasmania, you've probably got an awful lot of grasses, either ryegrass or other grass species that are carrying the virus and that are acting as reservoirs for aphids to pick up and then transmit into your crop. That becomes problematic because in terms of managing your inoculum source, that's really hard. If it came from the seed, you could do seed treatments, you could actually screen your seed, should say, and toss out all the infected stuff. But it's coming from the environment out there, and there's not much you can do about that. You can't spray out every grass within the 20 kilometres of your crop. No, it's just not practical. So the inoculum's there, the aphids are out there, they're probably picking up the virus, and then they're going to come into your crop. So now, what do you do? The epidemic in your crop depends on obviously the flight activity of the aphids, because they're the things that are bringing it, and virus sources obviously, depends on how much is there. Management. How do we manage it? Well, one of the things you can do is actually just understanding the biology of the aphids themselves. So with aphids, you've actually got distinct flight cycles. I'll probably get this wrong, but you know, like over summer they'll have a peak, and then they'll disappear down, and then they'll come up. So you'll have peak times when aphids are flying, and times when aphids are not flying. The populations are up. So they've done some work, and this is data I think from Tasmania too, where if you just alter the sowing date, so the date you plant, so depending if you plant early spring or late spring, early summer, greatly depends, and do nothing else. So all you're doing is altering the planting date, and do nothing else, you can greatly reduce the amount of virus you get. So early spring, you catch a flight of aphids that are coming in, carrying the virus into very young plants, and you get bang, you get a big hit of virus. Plant a bit later, and that aphid population's crashed down, and there's less flying around, and you actually end up with plants that actually have relatively little virus, just by altering planting date. Now, that depends on your capacity to alter planting date. Obviously, it depends. In Tasmania, we're kind of lucky, we've got a fairly wide growing window. Other parts of the world where you've got 10 feet of snow, you know, you've got fairly narrow growing windows, and you can't adjust your planting dates as well. Also, there might be requirements for market and harvest at certain times, in which case whether you can plant late to achieve harvest at the right time. Planting late, you tend to reduce, agronomically you tend to reduce your yield a little bit, but obviously if you're avoiding massive virus infection, that probably equalises itself out. The other big control for this particular virus is plant resistance. So resistance to the virus itself is really useful. If you've got a plant that's really resistant, that can have cellular systems that actually stop the virus infecting or stop it replicating because of a lack of recognition or some other factor in this plant cell, that's actually brilliant, because you can kind of ignore the virus from that point onwards. To date, with this particular virus, we haven't got resistance, we've only got tolerance. Who can tell me the difference between resistance and tolerance? Resistance is when you stop it. Resistance is a brick wall, saying, okay, plant's here, brick wall, virus is here. Can't get through. The virus is trying to get through, but hits a brick wall, can't get through. There's a defense process that stops it. But the pressure from the virus is, I want to get through, and it's going to keep chipping away, and eventually you might get resistance breaking, and it punches its way through the wall. It mutates enough to overcome that defense process and infects, and then you get disease. Tolerance is, you've opened the door, come in, that's fine. Infect me, that's fine. But don't cause damage. So cause very mild disease, not a severe disease. So you've actually got capacity of the plant to tolerate the infection without trying to restrict it. You still get some yield reduction, it's still not wonderful, but you also avoid the evolutionary drive of the virus to overcome a defense process, because it doesn't need to. It's happy, it's replicating, it's having fun, but it's not causing disease. To date, tolerance is all we have, and here's an example of tolerance. Don't know if you can see that at the back. These are barley varieties. Franklin has this YD2 gene, this tolerance gene. These are all infected. These are all infected with barley yellow dwarf. But these look pretty good, and these are going to yield fine. This is Triumph, and it's horrible, and it's not going to yield very well at all. Proctor is a cross between Triumph and Franklin. You can see it's kind of halfway in between. So this gene is actually really quite useful. Well, another story. The only reason Tasmania currently has a cereal breeding program, you think, we're not a big cereal state. We have one of the country's cereal breeding programs in our state. Why? Primarily down to this guy, Wayne Bertigan. So he was with state government up in Launceston, and just happened to have a really, really good green thumb in terms of serial breeding. So he was doing serial breeding at the time, and he was the one who successfully integrated this YD2 gene, which came from an Ethiopian line of something, wheat or barley or something. But managed to get this into a commercial barley variety, produced Franklin, and Franklin then became the dominant variety at the time, at least, for barley production. I'm not sure if it's still the case. It's a great malting variety. It was one that Kirin breweries would only use. So they would only, the big Japanese brewery would only source Tasmanian Franklin for their beer. I don't know if that's still the case, but it's a good story. Wayne's obviously retired now, but we still have a breeding program, a serial breeding program, based on the fact that we got a few successes and we're still doing that. YD2, however, provides tolerance for BYDV, barley yellow dwarf, but not CYDV, cereal yellow dwarf. So this is where the distinction between those two viruses becomes really important. Thankfully, CYDV is much, much less common here, so we get away with it. But understanding that, because if that situation changes, all of those Franklin and all those wonderful varieties of YD2 would become useless if CYDV became more important. CYDV is less important perhaps because the aphid vector is less important. We mentioned about insecticides. Insecticides can also be quite effective. It's easy to kill cereal aphids. To date, at least, there's no confirmed reports of resistance, although that might have changed. So insecticide treatments can be useful. Whether you use them or not depends on your economics. So obviously, if barley's got a good price in the market, you can afford to put a spray or two on. If you're getting a marginal return, then you've got to weigh up the cost versus the benefit of the treatment. And then you've got to think about timing. You can't, if it's a really, really valuable crop that you're trying to protect, it might be you spray it each week just to kill everything. You can't do that with a cereal crop. It's not worth that much. Your margins aren't that great. So you've got to time it when it's most important. So partly it's making sure you hit when the aphid flights are occurring. So if you're treating it when there's no aphids around, no aphid flights around, you're actually not going to get terribly much effect. Similarly, if you treat it too late in the crop growth, it doesn't matter because, as I said before, an older plant that gets infected will not have a great yield impact. It's the young plants that are infected that have the biggest yield impact. So your timing is protect the young plants, but also understand when the major aphid flights are coming. Because it's a slow virus to get in and get out, you can even have aphids arrive in your crop, start to feed, well, not quite, but you know they're coming into your crop. You can actually start hitting them with an insecticide treatment, one that's a systemic, that's going to sit there in the plant, so that when it does feed, and it takes an hour to actually feed, it's going to die before it transmits the virus. So you can use systemic insecticides that are throughout the whole plant, you apply them to the plant, so when the aphids arrive, they feed, they die before transmission occurs. You can apply systemic insecticides with the seed when you sow them, and so those very young plants are all dosed with insecticide at an early age and then are protected from virus infection until they get a bit older and are more likely to less likely to be damaged by the virus. This is in complete contrast to, as I said, those viruses that are non-persistently transmitted, those that just stick to the stylet, because they transmit in seconds. And those systemic insecticides in those instances can actually make the virus transmission worse, because the plant tastes horrible. It's got a poison in it, and the aphids come along and go probe, yuck, and then die. But meanwhile, they've transmitted about 5 transmissions with this really quick transmission. So knowing what you've got is really critical. Works for this guy, doesn't work for some others. Target major aphids, aphid flights, use slow release granules at planting. Understand the vector relationship with crop phenology. Where else? They have been playing with GE. We don't have that commercially, obviously, but here's a genetically engineered one that expresses resistance and works wonderfully. But even cultivation, because what we're dealing with here is aphids that don't like the aphid biology. Aphids love what we call a silhouette effect. So if you've got a green plant surrounded by dark earth, that's really attractive. And they just hone in on it, a bit like a target. If you've got a green plant that's surrounded by stubble, it doesn't see that differentiation and it's less attractive. So by direct drilling versus clean seed beds, you actually can, just by that, you can actually reduce your virus transmission. Okay. Didn't get time at all for these other ones, but you can have a look at those because they're interesting anyway. It's okay.
Yeah.